Good morning, everyone, and welcome to our holiday third Thursday for December. And welcome to Simone Castellarin, our presenter today. Dr. Castellarin is a professor at the University of British Columbia and a Tier 2 Canada Research Chair in Viticulture. Dr. Castellarin's research focuses on how the climate affects grape ripening and quality. In 2009, he received the Rudolf Hermans Prize for outstanding scientific achievements in horticulture and viticulture. His lab investigates the biological mechanisms that determine grape ripening and quality, and how these mechanisms are affected by environmental factors, temperature and water. The lab is developing viticultural strategies, irrigation, canopy and crop management, and hormone applications to adapt grape productions to climatic constraints and optimize ripening and the production of phenolics and aromatics in grapes. So with that, I'll hand it over to you, Simone, and welcome.
Thank you very much. Thank you very much. I hope you can hear me well. Thank you, Kathy, for the introduction, and good afternoon, everybody. I would like to mention that I'm connecting today from the UBC Vancouver campus, the Point Grey campus, who sits in the traditional ancestral territory of the Musqueam people. And today I would like to talk a bit about the effect of high temperature and heat stress on grape quality and general plant physiology. I will talk about some of our experiments that I will call in some cases preliminary experiments because they've been conducted in controlled environments. And then at the end of this talk, I'd like to mention some of the projects that we will start next summer and relate to the mitigation of high temperature in vineyards. So you have probably seen already some of these figures, and they come from publication, in this case, a publication that is now 10 years old, was considered controversial at the time, where the scientists started simulating the impact of climate change on wine regions around the world, and showed that in situation where greenhouse gases will continue to increase across this century, we might lose sustainability of grape production in some traditional regions of the world and improve the sustainability of this production in northern regions, including northern regions of Europe and northern regions of North America. Interestingly enough, some of the places where the sustainability of viticulture was forecasted to increase was the Pacific Northwest coast, and particularly the regions where we sit in British Columbia. So things are happening, things have been changing, and we are aware of that, and probably they will keep changing in the next decades. Going into the specific, I'd like to mention a more recent study that has been conducted by two scientists from the University of Toronto. And this study assessed the impact of climate change on grape growth and wine production in the Okanagan Valley. I must say that they used data from Kelowna, so the results of this study relate mostly to the Kelowna region and the Kelowna wine region. But these studies have assessed how climate and climate parameters that relate to grape production, including the growing degree days, average temperature, extreme heat days, etc., and precipitation. They have actually a dozen of parameters that have been analyzed, and they show how these parameters have changed historically from the 70s to current years. And also they model how these parameters are going to change in the future, like in 30 years from now or 70 years from now. Interestingly, what they observed is what I think several of us has experienced, that in the past 30, from the 70s, let's say, actually it's 40 years, the past 40 years, average temperature kept increasing. And the growing degree days accordingly have increased. So the study showed that there was an average increase of 2 degrees Celsius from the 70s to 2019. And these temperatures obviously relate to the growing season of the grapes from April to October. They also also analyze temperature during the dormant season, but I'm not going to focus on this, as today's topics focuses on heat stress and temperature effects during the growing season of the plant. When these scientists did some climate projection and tried to understand what we could expect in the Okanagan Valley in the next 30 years or 60 years, they use different models of predicting effects of climate change. They call one is the RCP 4.5, the other 8.5. I will just summarize by saying that one is a scenario, the 4.5, where greenhouse gas emission will keep increasing for other 15 years or so, and then will stabilize, versus the 8.5 scenario is a scenario that is quite extreme with continuous increase in greenhouse emission. So for just sake of time, we'll focus on the RCP 4.5 scenario, which is like intermediate scenario of climate change predictions. And what they forecasted is that in the Okanagan, the maximum temperatures, this is an average of the maximum temperatures during the growing season, will increase from 23 degrees Celsius, that is what is currently we are having, to 24.5 by 2050, and potentially to 25.15 by 2080. So we can predict to have higher maximum temperatures in the future, and up to 2 degrees Celsius higher in the next 60 years. Interestingly, they also analyzed the number of extreme heat days. So they analyze how many days during the growing season are above 32 degrees Celsius. They use 32 degrees Celsius as a threshold because several publications have used this as a threshold for indicating a very warm, hot day. I will say that I'm not sure if 32 is really a threshold we can use for predicting the behavior of the physiology of the plant. And I'll talk a bit more later about this. But this certainly indicates that is a good indication of how many really warm, really hot days we are going to have. And currently, we are talking about 27 days per growing seasons, where the maximum temperature is above 32 degrees Celsius. And in 2050, it's predicted to be 37. And 2080, 45 days. So basically a remarkable number of days during the summer that are above 32 degrees Celsius. And potentially, as we know from other type of studies and prediction, we'll have an increasing frequency of very hot days with severe heat waves with temperatures that obviously go to 38, 40, 42 degrees Celsius. So we know that the climate have been changing and is getting warmer. In some cases, having this warmer weather help us ripening the fruit more, depending of where we are and how warm it is. But it's definitely important to understand how these changes are going to impact the physiology of the grapevine and the quality of our fruit. We do know that vines are very sensitive to environmental factors. They could be water, light, temperature, as I say, and photosynthesis response to these environmental factors, and also the ripening related processes, accumulation of sugars, degradation of acids, accumulation of phenolics and aromas are very sensitive to the climate. And this is obviously well known. However, probably among the factors, there is somehow still a knowledge gap in the understanding of the effect of the temperature on the physiology of the plant and on the accumulation of metabolites in the fruit that are important for quality. The reason is related to the fact that it's not always easy to conduct experiments that simulate changes in temperature. We can do these experiments quite easily with some limitation in growth chambers. I'll show what is a growth chamber in a couple of slides, where we can control all the environmental factors inside the chamber where we grow the plant. But conducting such experiments in vineyards is complicated because it's difficult to manipulate temperature without affecting also, for instance, radiation and light in a vineyard. Some people have used heaters, or some people, well, most of the people have used heaters, but there are also, in this case, pros and cons in using them in the vineyards to simulate higher temperatures. So these are experiments that, generally speaking, are quite complicated and difficult to replicate. A lot of the information we have actually comes from experiments conducted in growth chambers. And I will talk about, summarize some of the things we know, and then present some of the experiments we have been doing to understand the impact of temperature and high temperature on the quality of the grapes. So generally we know that with increasing temperatures, with warmer temperatures, we accelerate the ripening process of the plant. And by accelerating the ripening process or favoring the ripening process, we tend to have more sugars, potentially more ripe aromas also in our grapes. This is true, obviously, but it's not necessarily always true, because we know that there are some temperatures where the plant, there are some thresholds that will actually impair the ripening process in the fruit and impair the accumulation of important quality compounds. We often refer to 35 degrees Celsius as one of these thresholds. We know 40 degrees Celsius is really detrimental for the growth of the plant and the ripening process. But in general, we know that when we are around 35 and above, from different studies, we learned that we have actually a decrease in berry weight, lower amount of sugars in the fruit, degradation of malic acid, and therefore decrease in titratable acidity, and also very strong effects, negative effect on the anthocyanin production in the fruit. This comes from a series of studies, as I say, that have been conducted both in growth chambers and also in some cases in vineyards. And here I am citing some of them. So the first study that goes, that relates to that we have conducted, and I would like to show today, is a study that Dr. Yifan Yan, a PhD student in our laboratory who recently graduated, conducted on Merlot grapes and published in 2020, and where she studied the effect of temperature regimes on the anthocyanin accumulation. As I say, those studies were conducted in controlled environment, what we call growth chambers. This is a good representation, a good picture of the chambers we use. We have several of these chambers in our laboratories, and we can put plants that are not obviously developed like a plant growing in a vineyard, but they are potted. In our case, we use unrooted grapevines. In this study was Merlot, and we have quite large pots of 12 liters, but the plant obviously is small in size, it's 3-5 shoots per vine. We put these vines, around 8 vines in a chamber like that, and we have multiple chambers. that we can use. And we grow these vines, in the case of these experiments, from variation to harvest at different temperature conditions. This experiment consider low temperatures, what we call low temperature, simulating like a cool climate, cooler climate, medium temperature, simulating a warm region, but not necessarily hot, and then high temperature that simulates a hot growing season. The numbers are here reported: 20 degrees Celsius day and 10 degrees Celsius night for low temperature, or 20 day and 15 night for low temperature. I'll explain why we have this also different night temperatures here. Medium is 25, 15. High temperature, as I said, was 35 degrees Celsius and 25 at 9, 35 and 30 degrees Celsius at 9. So in this experiment, we tested basically low, medium, high temperature during the day, but we also tested several differences in day and night temperature, because it's somehow believed that differences between day and night will favor the production of certain compounds in the grapes, including phenolics and anthocyanins. So in this case, we had treatments where we had difference of 10 degrees Celsius, and treatments where we had difference of 5 degrees Celsius between day and night. So I'll go straight to some of the results of this study, and it's quite clear how the temperature affects the accumulation of anthocyanins. The treatments that I just presented in the previous slides refer to the first year of study, and so are those treatments. The second year, we changed slightly the treatments, but the concept was the same: low temperature, medium, and high. And the data were very consistent. We ran actually this experiment for 3 times, and the data were always consistent. What we observe is then when we have low temperature during the day, 20 degrees Celsius, we have a much higher accumulation of anthocyanins than when we have a hot day. It is here on the bottom at 35 degrees Celsius. And average temperatures, like a warm weather, will have an intermediate level of anthocyanin. So this study has shown that actually anthocyanins tend to be favored in their accumulation in relatively cool weather compared to warm and hot weather. This is an effect that has been observed also by different other studies that show that anthocyanins are actually inhibited in this biosynthesis, but mostly degraded in their production if the plants are exposed to high temperatures in the vineyards and like in this case, in growth chamber. But another interesting result of this study was that when we change the difference between day and night in temperature from, as I say, 20 to 10 degrees or 20 to 15 degrees Celsius, so a difference of 5 or 10 degrees Celsius, we don't see any difference. So it's like the driver of this change in anthocyanin production is mostly the daily temperature, and this difference in temperature was not, between day and night, was not significantly impacting the accumulation of anthocyanins. Now, another effect that was interesting to observe, and we observe similar effects also when we apply other environmental stresses, is what we call the uncoupling between anthocyanins or some secondary metabolites from the sugar accumulation. So here in this graph, we have plotted all our data points collected from, as I say, grapes exposed to different temperature regimes, but with sugar level in the x-axis and the total anthocyanin in the y-axis here. So we look for a relationship between sugar accumulation in those grapes and anthocyanin accumulation. And here you can see that growing grapes at high temperature uncouples the production of anthocyanins from the accumulation of sugars. What it means is that, for instance, if you take a fruit at 20, apologies, trying to have my pointer here. Yeah, at 20 bricks, you'll have very low level of anthocyanins if the vines or the grapes have grown at high temperature, or remarkably higher levels if the grapes have grown at 20 degrees Celsius. So this uncoupling is interesting because it tells us that, well, sure, sugars are a very important indicator of ripening, but we cannot always use them as a proxy for the amount of anthocyanins or other secondary metabolites like aroma. It's not shown in the study, but other studies have indicated that. So we cannot always use sugar as a proxy for the amount of these other quality compounds. So the other study I'd like to talk about that was conducted more recently by another collaborator, Dr. Joshua van der Weide, and myself, was the impact of heat waves. So how heat waves are affecting leaf physiology and berry composition. Heat waves are obviously one of the challenges that wine regions are experiencing around the world. And we had some experience with heat waves here in British Columbia, particularly in 2021, where this study was conducted. And interesting enough, our simulation of heat waves that I thought they were quite dramatic for the grapevine really matched, like I'm going to show in a graph later on, the heat waves that vines were experiencing in the Okanagan Valley the same season. So heat waves, before I go into our data, are these, consider these short period of times, few days, 3, 4, 5 days, with extremely high temperature or higher temperature than the usual. They have been studied in viticulture, not extensively, but there are now a good number of studies that are showing what can happen in a grape when it's exposed to heat waves. Obviously, being the number of studies limited, we have knowledge about some varieties, but not many varieties, and not necessarily all the varieties will have the same sensitivity to heat waves. However, I'm showing this graph and this slide from a study conducted in Australia and published in 2010, where they simulated heat waves in growth chambers on Semillon. The heat wave was about 40 degrees Celsius during the day, 25 degrees Celsius at night for 4 days. And what is interesting about this study is that they subjected this grapevine to heat waves at different stages from pre-veraison. Actually, they started the blooming, but they had a heat wave at blooming, heat wave pre-veraison, then veraison, and then mid-ripening. I'm not presenting all the data from the study. I'm just highlighting a couple of things. The first thing is that the impact of a heat wave on plants that on the photosynthesis, so on the capability of the leaves to take carbon and accumulate and produce sugar. That sugars go distributed across the plant, but particularly in our case, because we are interested in the ripening, also go in the fruit and accumulate in the fruit during the ripening process. So an interesting, those are, in this graph, we have the net photosynthesis of grapevines that are growing in normal conditions. For them, normal conditions where I don't remember exactly, between 25, 30 degrees Celsius during the day and 15 at night. But then when they expose these vines to a heat wave of 40 to 25 degrees Celsius day and night, you see that photosynthesis strongly decreases and stays low for the days of the heat wave. And then it takes several days to recover when the plants are then exposed to more normal temperatures. And what they observe is actually these effects of heat waves on the berry fresh weight and the soluble solids or sugar content change depending on when the heat waves were imposed to the vines. And they observe stronger effects on photosynthesis, but also stronger effects on the berry size and sugar accumulation when the heat waves were applied at veraison or during the ripening process. And these effects were negative. So when the plants were exposed to the heat wave, they had it was observed a reduction in the berry size and also a lower amount of sugar accumulated in the berry. And this is similar to what we found in our experiment. Our experiment was conducted in, as I say, in 2021, and it was considering the application of a single heat wave on Gewürztraminer vines. The single heat wave was applied just after veraison, so during the ripening process, or a double heat wave, where plants were subjected to a heat wave before veraison and a heat wave after veraison. What is a heat wave for us is it was like plants that were exposed to 25 degrees Celsius during the day and 25 degrees Celsius during the night and 40 degrees Celsius during the day. And they were compared to milder condition like 20 degrees at night and 30 during the day. So those were vines that were already growing in in a warmer climate, but they were exposed to a sudden increase in temperatures. And interestingly, these are the data from June 29, 2021, Penticton, and you can see that the temperature values 20 to 40 were not far away from the temperature that we were simulating in our growth chambers. So one thing is when we expose plants to heat waves, we assess on a daily basis how much water the plants were needing or were using. And we were replenishing the very same amount to the plants, meaning that we never let the plants experience any stress. And we observed that by exposing these plants to a heat wave, the irrigation that we had to apply was 50% higher during the single heat wave, and 30 or 80% higher during the double heat wave, depending if we were considering the pre-veraison heat wave or the post-veraison heat wave. So we had to apply way more water to these plants where they were exposed to higher temperature. And we observed the same thing that people have observed before, that a heat wave reduces photosynthesis, regardless if it is applied as a single heat wave or a series of two heat waves, pre-veraison and after veraison. However, when we had a double heat wave applied to our vines, we observed that there was a recovery, like has been shown before. The plants, they shut down photosynthesis, but then they recover. But we observed that these plants that were exposed to two heat waves had reductions in photosynthesis also later on during the ripening, particularly at harvest time. And this indicated that the double heat wave somehow hit the plant in a way that could not recover to normal values. Another thing that we learned from this is that, yes, by irrigating, applying more water, we cannot completely mitigate the effect of the heat wave. Applying more water during a heat wave or during during days with high temperature is a common practice and helps the plant mitigating the stress of a high temperature. However, it does not prevent the plant from suffering, let's put it this way, suffering and decreasing photosynthesis during heat wave. An effect that we also observed in this study was that the single heat wave reduced berry weight, but not the double heat wave. And the double heat wave reduced the total soluble solids in the sugar. And that was consistent with the fact that double heat wave did not allow a complete recover from the heat stress situation versus the single heat wave after the decrease in photosynthesis for the 4 days of the heat wave, then the plants recover completely. Another interesting observation of this study that we did publish is probably less important in terms of impact on the quality of the fruit, but probably very important in terms of the understanding of how the plant responds to this situation, is related to another analysis we did, prompted by this intriguing result that we found on the berry weight, where the berry was decreasing in weight with a single heat wave, but was not decreasing in weight where we were applying 2 heat waves. And that looks strange to us because 2 heat waves should have a stronger effect than a single heat wave on the berry growth. And I will save you most of our data because, as I said, probably is not so much interesting for an applied perspective, but certainly interesting from a physiological perspective. What we observe is that double heat waves change the profile and the amount of waxes that were produced and accumulated in the on the top of the berry. So probably you're all familiar with fruits and grapes, and you know that there is a waxy layer that is accumulating these berries. This waxy layer is very important to protect the fruit from water loss and the transpiration, so the loss of water from the fruit. We know that some environmental factors change this waxy layer, and including water stress, etc. But we observed that these heat waves were changing this waxy layer by increasing the amount of waxes and changing the composition of these waxes. And accordingly, the fruits were losing less water when we were testing how much water they were transpiring. I'm not going to go into the details of this graph, but the take-home message is that fruits that were exposed to double heat waves had more waxes and lost less water through the cuticle, through the surface of the fruit. And this probably helped the fruits withstanding the effects of the heat wave, and therefore there was no difference in the berry size. I think this is something that we will study more in the future, because in the same year, 2021, when we had a heat wave, some observation by scientists in Oregon and Washington State on blueberries, where waxy layers are very important for quality, observed that heat stress and the heat wave were changing this waxy layer remarkably. So we will follow up in the future to better understand what is the role of these waxes that have no impact on the quality of the wine, but might have an impact on how the berry withstands the heat stress in the fields. So another experiment that I want to briefly mention is what I call a preliminary experiment, because it was run only 1 year, and we would like to run for multiple years in growth chambers before we have a conclusion. But this experiment aims to understand what happens when a heat stress situation occurs in combination with a water deficit situation, like water scarcity. Is this interaction important, relevant? What is the best situation for growing our grapes? Do we want well-watered grapes, or it doesn't matter? In this case, we work on Riesling, and I'm gonna present only data that are related to the effect of these stresses, water stress and heat and high temperature, on the aroma, and particularly the terpene compounds, those floral or fruity aroma that we find in certain varieties, including Riesling, Viognier, Muscat, and Gewürztraminer. So in this case, this experiment was conducted again in these growth chambers where we could modulate the temperature. We had 22 degrees during the day and 12 at night, versus 2 other chambers where we grow plants at 32 degrees Celsius and 12 at night. So these are 4 chambers where we had in 2 chambers, let's say, warm weather, but simulation, and the other hot weather simulation. And within this chamber, we normally can accommodate 8 plants. And we had half of the plants that were well irrigated and half of the plants that were exposed to water deficit situation with irrigation managed at the 30% of evapotranspiration. And as I said, we did collect multiple type of data, but the most interesting for us, for our goal was mostly to understand the effect of these conditions on the aromas, particularly on the terpenes. In this case, we are reporting total free terpenes, so those volatile compounds that are released by the grape or by the wine, and strongly contribute to the aroma of Riesling and other varieties. So in this experiment, we observed that in most of our treatments, 3 out of 4 treatments, the accumulation of terpenes increased during ripening. This is day after anthesis. So this is basically when we started the experiment, and we put the plants in the different conditions. And we observed that well water and or water deficit plants at high temperature had low terpene levels. The same was with low temperature water deficit. The terpene levels remained low. What we observed was that at low temperature and well water plants tend to favor the maximum accumulation of terpenes. Now, this is, as I said, a sort of a preliminary result, because we need more validation with different multiple experiments conducted before we can confidently say that we need to have well water plants and low temperature maximize the terpene production, but is an indication that what we suspected before, that is that there is an interaction between temperature and irrigation levels, and with irrigation and temperature will interact, and therefore understanding how to manage irrigation in different conditions is important. Yes, to withstand the physiological response of the canopy to a stress, but also to determine the quality of our fruit at harvest. So this is not the conclusion of the talk. I still have 5 minutes, but is the conclusion of the first part of the talk, that is based on what we learn in these experiments. So temperatures, if they are high, they negatively modulate anthocyanins. So hot temperatures also negatively modulate terpenes, but it needs to be confirmed. Heat waves, well, have negative effects on photosynthesis, sugars, and berry size, depending if they are 1 or 2 in combination. And this physiologically interesting response of the waxes to the heat stress, that might be one of the mechanisms that the plant have, or the fruit have, to mitigate heat stress effect. But what we do need, going more toward the application of this information, is to develop mitigation strategies in vineyards to cool down these canopies that if they are exposed to very high temperature. And also we need to better understand the response of fruit quality to temperatures. As I said, we know more about anthocyanins, but we know not much about the impact of these temperatures on the aroma of the grapes. And here in the last 5 minutes, I just want to mention a project that we are going to start this summer in vineyards and in growth chambers to understand the temperature and heat stress mitigation strategies that we could use in vineyards to mitigate potential heat waves or high temperatures that could happen in certain seasons or for part of the season. This is a collaboration between my laboratory and Dr. Nathaniel Newlands at Agricultural Food Canada, the Summerer Station. And I believe Nathaniel might be in this call, so if there are questions related to that part, maybe we'll jump in and answer. But I'll present the major goals of these topics, of this project, excuse me. So the first goal is to conduct vineyard experiments to understand whether varying irrigation can help us mitigating the response of the grapevine to heat stress. We do know there will be a response, but we are curious to see if these various irrigation level will really affect how the plant will stand heat stress in terms of the quality of the fruit at harvest. The other is to assess the capability of biostimulant and anti-transpirant to mitigate heat stress and heat waves in vineyards. So the application of treatments that I will explain in a minute to cool down or help the plant withstanding heat stress in the vineyard. Then growth chambers experiments where we want to understand how the different varieties, and we will consider major varieties for BC viticulture, withstand heat stresses. Are some varieties more sensitive than others, and which one they are? This might help understanding how a given variety in a given condition will respond or what we can expect from them, and maybe take some educated decision when it comes to planting a given variety in a specific spot that is very could get very hot during the summer. And finally, the part of the project is the one that is led by Dr. Newlands, that is conducting a regional scale study of temperature effects on grape quality, particularly sugar, acids, and aroma in the Okanagan Valley, and model optimal environmental condition for grape quality in short-term and climate change scenario. Basically build some models that can predict the amount of quality-related compounds in the fruit in different temperature scenarios, including increases in temperatures that we can forecast in the future. I will briefly present this project by saying that the first one is where we will modulate irrigation and expose plants to well water situation, or even increase water levels compared to the water they use, that we call evapotranspiration of the crop. But we'll compare this with situation where water is plants are exposed to moderate water deficit, which is quite common in several vineyards when it comes to produce red grape varieties like Cabernet Sauvignon and Syrah. This experiment will be conducted in the southern part of the Okanagan. We haven't identified the vineyards yet. Actually, if you have interest in contributing or collaborating, I should be saying, in this project, we are very welcome to discuss with you potential trials in your vineyards. And in this case, we'll assess when hot days will occur and manage the irrigation in relation to these days, and then assess how this difference in irrigation manager will impact the quality of the fruit at harvest, including also other parameters related to the physiology of the plant. The second study aimed to consider two other varieties, even though we haven't decided exactly which variety. We thought about Merlot and Gewürztraminer, but we still have some months to identify which variety exactly to target. We thought Merlot because it's a very important red grape for BC, and Gewürztraminer because it's white grape that have high level of aromatics, and we would like to capture the effect of these on treatments on aromatics. But the idea is to use strategies that people have shown to help mitigating the effect of temperature on the canopy, including kaolin, zeolite, which is a new product that I don't think is commercially available, but has been tested in different vineyards in Europe, to help cooling down these canopies exposed to high temperature, or chitosan to stimulate and protective responses in the cells, as well as anti-transpirants that have been shown in Australia to help mitigating the accumulation of sugar under high temperature conditions, but also mitigating the effect of heat waves. So those are not published data, but have been communicated in different magazines and articles. So we are curious to see if or to investigate also the potential mitigating role of anti-transpirant. As I said, we will also test in the third project how different grapevine varieties withstand very high temperatures and how quick they recover from such a stress. In this case, you can see here multiple varieties that we grow in British Columbia, and it will be done in using potted grapevines in a different physiological stages, like fruit set, lag phase, variation, and ripening. And in all these projects, obviously, we will collect physiological data and temperature data, leaf water potential, etc. So we'll characterize how the canopy is doing, but also will characterize the quality of the grapes, including volatiles, aroma, and phenolics. Finally, the last slide before the acknowledgement is that this is the part led by Dr. Newlands and in collaboration with our laboratory, and is where we will conduct this regional scale assessment of the impact of temperature on fruit quality and yield, etc. And this will be done by looking at different regions of the Okanagan Valley with different temperatures, assessing multiple vineyards for each region, and also multiple locations within a vineyard to understand how the various climatic factors, temperature is a major one, but also water availability and soil condition, etc., affect the production of or the accumulation of sugars and aroma in the berry. Dr. Newlands will lead this machine learning modeling to understand the role of these climate variables. It will integrate multi-scale historical and future climate information to predict, basically, to build prediction models on how temperature and other factors can affect the quality of grapes, particularly in scenarios with increasing temperatures and heat stress scenarios. So with this, I would like to acknowledge, and I'm sorry it took 3 extra minutes than planned, but Dr. Yifan Yan and Joshua van der Weide, who have performed the growth chamber experiments that I presented today, Dr. Nathaniel Newlands, who is collaborating with us in developing these new experiments and new projects, the BC Wine and Grape Council, who has always been supportive to our laboratory and will support these future studies, as well as NSERC and for their Discovery and Canada Research Chair program, who have funded part of the results, experiments that I presented today. So thank you very much. I'm happy to take any question. Thank you.
Thanks, Simone. I think it's best if questions get popped into the chat because there's quite a few people here. Simone, the Wine Grape Council is really happy to continue to be able to continue to support this work, but you're going to have to update our logo on the acknowledgements page.
Okay.
Because we're really proud of our new logo.
Oh, yeah. Sorry about that.
No, that's good. I mean, it's it's fun to see the past one as well. I imagine there's some concurrent work being done on heat-resistant rootstock. Am I right about that, or just drought-resistant rootstock? Because I see you chose to use own-rooted vines.
Yeah, okay. So that's a good question. The reason why we use on-root, I like just to clarify, we use on-rooted vines because, well, this is for us is a way of producing those grapevine for our growth chamber experiments. We get clean material from Sydney station, so we generate. Every year we generate new plants that then we use in the experiment the following year. We can also run and purchase, varieties with grapevines with different rootstocks as well. We haven't worked on assessing the impact of the rootstock. I know that I'm not aware of studies on the assessment of the heat stress on rootstocks. I know I was probably they are. I know that I was we were considering and proposing some of these studies in collaboration with colleagues at Brock University that are considering the impact of rootstocks and scion combinations on different factors, including climatic stresses. So I don't know. I don't think we have specifically rootstocks that are temperature adapted. I don't think that was a major goal for their selection, but it is possible their performance, particularly in how they affect the canopy as well, so the upper part of the vine, the rootstock might affect the response of our scion, of our variety to the environmental conditions. So that's certainly a possibility that rootstock have an impact.
So if someone were to volunteer their vineyard to be part of your study, would you prefer that they be own-rooted vines?
No,
Doesn't
no.
matter.
And actually, in our proposal, no. In our proposal, we indicated several rootstocks. The rootstocks are mostly used in the Okanagan. Depending on the study, we would like to have consistency. So if we have multiple vineyards, we will look for the same rootstock just to avoid to introduce another factor. It will be very interesting to have to taste different rootstock. But the problem is then you introduce another interacting effect, and that might be a confounding effect on what we are trying to analyze. But definitely we are fine, and we were planning to use vineyards with rootstock, with major rootstocks that are used in British Columbia.
Thank you. Do we have any other questions? You must have been very clear, Simone.
Or maybe very boring, but anyways.
No, we've got a question, Lisa Lombold. Have you thought of including anti-transpirants such as Laustim Osmo, which is glycine betaine, and anti-stress, which is 44% acrylic polymers? She says she's had good anecdotal feedback from a grape grower who had trialed Laustim on half of a vineyard block this season. They had major irrigation break with no watering during this period. Where it was applied, the vines appeared significantly less drought stressed.
Yeah. So this is thank you, Lisa, for the question. And as I said before, when we still have for I guess 6 months before we'll start our experiments next year. And we are still considering, well, we are discussing and further discussing with the industry and potential partners, our wineries, or on what treatment to test. We do have, as I said, an idea about which one we wanted. We wanted chitosan, we wanted kaolin. We thought about zeolite, but I'm not sure if it will be possible to find commercial zeolite available. We would prefer treatments and applications that are commercially available so that the growers later on can access them, and including the anti-transpirant. We indicated was the one that we knew was commercially available. But if certainly there is still room to reassess some of these treatments to see if we can include some new products that are considered potentially helpful for mitigating these stress effects. And I'll be in contact. Hopefully we can stay in contact and discuss, and because I just would like to add only one thing. Some of this information, there is not much published on some of these biostimulants. We have some data, but not so so much. Often is it comes from some experiment conducted in vineyards, but didn't end up being published in scientific papers. So there might be some products that are valid, but I am not aware, and it's worth investigating.
I have a question. Thank you. I have a question from Michael Clark. He says, you showed that the double heat wave experiment changed the waxy layer on the berries, reducing transpiration. Was the point there not specifically that it was double heat wave, but rather that the first wave was pre-beraison, i.e. is the heat wave pre-beraison key to the change in the waxy layer?
Yeah, so that's a very good question. Yeah, I agree that could be that the reason, and I should have mentioned. So my point, honestly, my point with that, showing you that result today, is not to specifically say one or two for that specific context. It was an experiment we ran after observing these in the double heat wave. And this is a really valid point, that probably what was triggering this change in wax amount and wax profile or composition is the fact that the heat wave occurred early on during the pre-veraison part of the season. And probably that helped them withstanding the second heat wave or whatever happened later on. So I agree that we cannot rule out one or the other, but probably this is what is happening. And I'm mentioning this is an interesting point for future studies on the role of these waxes in protecting the fruit from stresses.
Is there modeling to show that if we have one heat wave, we're likely to have a second in that season?
Can you repeat? If we have, excuse me?
Is there modeling, like climate modeling, that shows that if we have a heat wave, we're more likely to have a second one in that season?
So I haven't conducted any climate modeling. I know that, so in the paper I assessed I was presenting about the BC climate, I don't recall them talking about heat waves and frequency, but it's quite how to say, is believed there are several publications showing the frequency of heat waves will be higher, including, I think, the frequency within a season. But I have no, honestly, I'm not sure about that, in the sense that I don't have information for these.
Right. We need Dr. Volkovich back.
Yeah, exactly, Dr. Volkovich and.
Okay, I want to thank you for being here. Of course, we have one more question.
Okay.
From Benman, which before we leave, I want to mention that our next third Thursday is January 18th next year, and it will be Benman Chang from the Research Center in Summerland, and he's going to talk about the current status of the work of using imaging to diagnose grapevine trunk disease. And his question to you today, however, is, will you measure the wax composition on the berry skin and leaf surface change before and after the anti-transpirant application?
Oh, okay. So first of all, briefly, I'd like to say hi to Ben Min and acknowledge that he is conducting, and he's also developing techniques to mitigate heat stress, if I'm not mistaken. So I hope we can collaborate within this project. And but I was not planning to. This is a very good question. If you're referring to the future study, if I'm planning to measure the waxes, I think it would be a very interesting thing to do, because the anti-transpirant, how is advertised, adding further coating and protecting from dehydration the berry. I'm curious what happens on the waxy layer produced by the plant. Yeah, so I think that we will add as a side project on this treatment some analysis of waxes to further understand what is the response of the fruit or this part of the fruit to our treatments.
Okay, thank you, Simone. Thank you to everyone for coming. We'll see you next year. I hope everyone has a great holiday. And thanks for your support of the BC Wine Grape Council and the R&D committee. Okay.
Thanks, everyone.
Yeah.
Thank you, Kate.
Happy holidays. Thanks,
Happy
Simone.
holidays to everybody. Thank you very much for attending.
Thank you. Bye.
Bye-bye.